Hello, I'm Dr. Crystal and Morrison for Wayne County Community College. Today, we're going to study the eukaryotic cell and just learn a little bit about eukaryotic cells and the microorganisms and some other microorganisms. So, we've already learned quite a bit about prokaryotes and all the different kinds of bacteria they represent, like mycobacterium with supplying TB and causing TB and leprosy, streptococcus, staphylococcus, mycoplasma, known as that causes walking pneumonia, the spirochete that causes syphilis. We learned about obligate parasites that require a host, like rickettsia and chlamydia. We learned about gram-positive and gram-negative and acid-fast staining. So we've learned about the makeup of these bacteria and their vulnerability so that our antibiotics can attack them and defeat them and make us eukaryotes healthy again. So we learned a lot about prokaryotes. So let's learn about this huge group that we belong to and learn about even some other eukaryotes. So it's not just prokaryotes or bacteria that want to harm us. Sometimes even eukaryotes want to harm us. These will be known as the parasites, especially worms known as helminths or protozoans, these little microscopic animals like the amoebas. They're seen in most other parts of the world, usually in the tropics, and they can cause debilitating disease and disfigurement. Some scientists say that the eukaryotic cell was developed due to symbiosis. Here it is here. Sym meaning together, biosis or bios meaning to live or live. This close association between these two organisms, when a larger prokaryotic cell engulfed several smaller prokaryotic cells. And somehow this protected the smaller cells, right? And the smaller prokaryotic cells got together and they began organizing themselves. And these other cells became specialized and began to take care of all the nutritional requirements like oxygen and metabolism, etc. So now this made this a symbiotic relationship where the prokaryotic cell that engulfed, the larger prokaryotic cell that engulfed all the smaller prokaryotic cells can now have a symbiotic relationship and have an interdependence. And this mutually beneficial relationship allowed the cell to be more versatile and increase its chances of survival. Over time, this existence evolved into a relationship that became obligatory. In other words, one cell was obligated to the other. It was now necessary for the larger cell to survive, and it needed all of these things that we now call organelles. And that is how we think that eukaryotes were formed, from a series of smaller prokaryotes after they were engulfed by a larger prokaryote. And the relationship became symbiotic, where they could live together and benefit in a mutually beneficial way. So the eukaryotic cell are protozoans, algae, fungus, and animal cells, right? That is what they would consist of. The flagella in eukaryotic cells, remember flagella has to do with motility, right? It's found in bacteria, protozoans, algae, fungus, and some animal cells. The flagella that we know for in humans is with the sperm, right? Cilia also is for motility, and that's found only in protozoans and animal cells. And in humans, we have cilia in our respiratory system and in our reproductive systems, little tiny hairs that beat. to and fro, right? So Remember, the flagella in the eukaryotic cells, though, is a bit different. The flagella in eukaryotic cells whips back and forth, as we discussed previously. An example I gave then again too was the sperm, human sperm. As opposed to the prokaryotic cell, which the flagella goes in a 360-degree circular motion like a propeller, right? All right. Let's look over here real quick since I mentioned the cilia. Okay, so that was flagella due to motility. Eukaryotic and prokaryotic flagella move in a different way. The cilia, though, are microscopic little hairs that beat, especially in our respiratory system, to keep mucus and other particulate matter out of the lungs, for example. Here's a cross-section of cilia. I want you to notice these things called doublets, right? You have 9 doublets in this cross-section of cilia. And you have these 2 singlets here in the middle. This is a 9 plus 2 arrangement of microtubules. So this is a microtubule or cilia that's cut cross-sectional and lets you see what is actually going on to make the movement. And the way these cells move, the way these cells are arranged, allow for movement of this microtubule. So it's 9 plus 2 arrangement of microtubules makes movement possible. And that is for the cilia. Particular arrangement of cilia is in our respiratory tract, and I said also in our reproductive tract. And we have to remember that they use dynein arms to help all of this move. What's the big deal with motility? Well, motility, did I write that anywhere? No. So motility allows a microorganism to locate life-sustaining nutrients and to migrate toward more positive stimuli, like maybe the sunlight, for example. It also permits avoidance of harmful substances and stimuli. So motility is really a good thing to have evolved to. All right, let's look at the glycocalyx. The glycocalyx is comprised of polysaccharides, and we know polysaccharides means many sugars. It promotes the adherence to environmental surfaces and the development of biofilms. Remember, we learned about biofilms before. So the glycocalyx is similar in eukaryotes and prokaryotes. You know, it's a slime layer. And sometimes it could be on the prokaryote, it could be a covering of on the capsule of a prokaryote. But it's a network of fibers that promote adherence to environmental surfaces and the development of biofilms. Receptor, communication, and protection from environmental changes. So I need you to remember that. So this will be an important thing. And one thing that glycocalyx, like a biofilm for us, would be particularly plaque or something on our teeth. We see it in showers, the scum stuff on showers, etc. That's actually for bacteria, though. So we'll just keep it as this for the eukaryotes. It is an adherence, a glycocalyx, so you can tell it has a sugar coating in it, plus it tells you here it's composed of polysaccharides. Let's look at the cell wall. Cell wall. Cell wall is the outermost layer of plant, fungi, and bacterial cells. It adds support for growth. It's made of a tough cellulose, and it's hard to digest. Just think of something like celery. That's the example I have here of something that has a tough outer layer and difficult to digest. The cell wall for fungi and algae are rigid and provide structural support and shape, right? See if I have anything else. So remember, this is for eukaryotes, which animals belong to. You'll remember that here. Animals do not have a cell wall, along with some protozoans and a few algae. Some of the algae, though, will be encased in a cell membrane, but they may not have a cell wall. Fungi and most, but not all algae have a thick, rigid cell wall. So fungi, but most algae, but not all, have a thick, rigid cell wall surrounding a cell membrane. And remember, that helps to add structure. Algae cell walls vary in chemical composition, so they have different chemical compositions that algae does. Cellulose, pectin, mannans, and minerals, like, for example, silicon dioxide and calcium carbonate may make up algae cell walls. That's very different. Fungal cell walls have a thick inner layer of polysaccharide fibers composed of chitin, C-H-I-T-I-N. I'm going to ask that about fungus because that's an interesting type of cell wall. Or they may be made of cellulose and a thin outer layer of mixed glycans. So anyway, it's a lot of sugars involved with the cell wall. Right? So make sure you kind of remember that chitin is the cell wall for fungus, right? Let's look at, let me mention this. Mitosis of a eukaryotic cell starts with interphase. Interphase is before mitosis. And then you have the first phase, prophase. So we'll put this as 1, metaphase 2, anaphase 3, telophase 4, and then this whole thing was making the cytoplasm is cytokinesis, right? Okay, so the process of dividing the cell contents to make 2 new cells. So that's what this whole mitosis is. It creates 2 identical cells from 1 original cell, right? So it takes a parent cell and makes 2 identical cells from that parent cell or the original cell. All right, let's see. Do I have something here? Let's look at the nucleus. The nucleus is the control center of the cell. It has these pores or these narrow spaces that allow for certain molecules to selectively migrate between the nucleus and the cytoplasm. The nucleolus is a granular mass, and it makes up the main body of the nucleus, along with a matrix known as the nucleoplasm. The nucleolus is the site for ribosomal RNA. So this is the site for rRNA, okay? And that's where the synthesis and a collection area for ribosomal subunits occur here in the nucleolus. The subunits are transported through the nuclear pores into the cytoplasm for a final assembly into ribosomes. Ribosomes for us, let me see if I'm going to get to that. I'm pretty sure because ribosomes for eukaryotes are pretty important, and I'll get to it, but I want you to remember that they're 80S subunits, right? 80S. So the nucleolus is the place that ribosomes are made, then they push out through the nuclear pores from the nucleus, okay? Chromatin, here's chromatin. Chromatin stains darkly and comprises the chromosomes, right? Chromosomes stain dark, and they really make up the chromosomes, and that's the genetic material needed because the eukaryotes will have genetic material to transfer to future generations. Endoplasmic reticulum, known as ER. You have a rough endoplasmic reticulum, known as RER. And you have a smooth endoplasmic reticulum known as SER. So the endoplasmic reticulum, the rough endoplasmic reticulum originates from the outer membrane of the nuclear envelope, right? Or the cisternae, you could call it, to serve as a passageway from the nucleus into the cytoplasm. So from the nucleus into the cytoplasm, That's what the rough ER acts as this passageway. And ultimately to the cell's exterior. On the other hand, the smooth endoplasmic reticulum has a similar function in the synthesis and storage of transport, but mainly the smooth ER is storage and transport, but mainly of non-protein molecules such as lipids. So when you think of smooth, think of something that's fatty or like lipids. It also serves as a site for detoxification of a variety of metabolic products. So the smooth ER is there for serves as a storage site for lipids, for transport of lipids, and also a site for detoxification of a variety of metabolic products. Okay? Let's look at the Golgi apparatus, also known as the Golgi complex. This is the site in the cell where proteins are collected and packaged for transport to their final destinations. Some vesicles or sacs contain enzymes to digest food inside the cell. Other vesicles are secreted to digest materials outside of the cell, and others are important for the enlargement and repair of the cell wall and the membrane. But whenever I think of Golgi apparatus or Golgi complex, I want you to think of it this way as well. The proteins are chemically modified and packaged into vesicles or little sacs to be used by the cell in a variety of ways. So think of Golgi as a packaging center, packaging and kind of a distribution center. Okay? The lysosome contains digestive enzymes, breaks down food and kills pathogens. And autolysis, auto meaning self, and to tear or rip, destroy dying and aging cells. That's another thing that lysosomes do. Lysosome is a type of vesicle originating from the Golgi apparatus. So the lysosome comes from the Golgi apparatus, and it contains a variety of enzymes. They're involved in intracellular digestion of food particles and in the protection against invading microorganisms. They also participate in digestion and removal of cellular debris in damaged tissue. So that's what lysosomes take care of. Lysosomes also, because we have some, they are natural defense against bacteria by hydrolyzing the peptidoglycan in the cell walls. And we learned about peptidoglycan last time. We know that certain antibiotics can attack the peptidoglycan in the prokaryote or in the bacteria and kill it. But we too have these lysosomes that can attack that peptidoglycan cell wall and kill that bacteria that way. So if you think about lysosome, just think about it having the enzymes that are there to help protect the eukaryote against invaders. Vacuoles. Vacuole stores food and water waste and has a color pigment. Usually it's the largest part of a plant cell, right? So vacuoles are membrane-bound sacs containing fluids or solid particles to be digested, excreted, or stored. They are formed in phagocytic cells, like certain white blood cells and protozoa, in response to food and other substances that have been engulfed, right, or eaten like that. A phagosome is a merger or combination of a lysosome and a vacuole, something that's going to come around and eat something up. So that's what a phagosome is. It's the merger or combination of a lysosome and a vacuole. Okay? We're going to come back. So that is what I have for now. We're going to come back and finish up on some other definitions, because all of these things that we're talking about are organelles, and each little thing in the eukaryotic cell, and this is mainly like I'm talking about humans now, well, all of them, have a specific duty. So I'm just going through listing, just giving a synopsis of the duties of each organelle in the cell. And you'll be able to compare that to what a prokaryotic cell looks like. But the eukaryotic cell has a lot of organelles and each have a different function. So we'll come back and look at the rest of them when we get back. Okay, let's talk about mitochondria here. Mitochondria has two major parts to it, right? The cristae, which are the folds on the inner membrane that hold the enzymes and the electron carriers of aerobic respiration. This is an oxygen-using process that extracts chemical energy contained in nutrient molecules and stores it in the form of high-energy molecules, or ATP. So whenever you think about mitochondria, it would be a great idea to think of it as an energy source where you get your ATP from. That is in the crista. Now another part to the mitochondria is known as the matrix. The matrix are the spaces around this cristae, or crista, and they're filled with a chemically complex fluid. This fluid holds the ribosomes, the DNA, and a pool of enzymes and other compounds involved in the metabolic cycle. Now, interesting thing about the mitochondria, we already know that it provides energy to the cell. That's the ATP, right? I want you to know that there's another interesting thing called endosymbiosis. And there's an endosymbiosis theory about mitochondria. The theory is that they were once free-living organisms that became part of modern cells. The evidence of this is that they have their own DNA and ribosomes, and they make proteins and can reproduce and replicate. So that would be an idea or an example of endosymbiosis, right? So let me go on and talk about the chloroplast, and then I'm going to show you how the mitochondria and the chloroplast can be great examples of endosymbiosis. So chloroplasts are found in algae and plant cells. Notice, animals do not have chloroplasts. Please make that a point of knowing that. Chloroplasts are found in algae and plant cells. They're capable of converting the energy of sunlight into chemical energy through something we learned in 3rd grade called photosynthesis. So I'll just say animals get their energy source from mitochondria. Plants and algae get their energy source from chloroplasts. Now something else interesting about the mitochondria in humans. Humans only inherit their mom's mitochondria. In other words, if you share the same mother with your brother or sister, then you guys have the same mitochondria that she got from her mom, that her mom got from her mom, and her mom got from her mom. On and on, all the way back to whatever you believe in, Eve, for example, if you believe in that. So that mitochondria is handed down all the way through a matrilineal lineage from that, or not lineage, but inheritance, and that comes from each mom. So your great grandmother, you have her mitochondria because it came through all the way to your mom, if it's on your mom's side. So you and your brother, you and your sisters, if you have the same mom, you have that mitochondria dating back for centuries, right? So I think that's interesting to know. The other thing is, let's see. So let's talk about what's unique to mitochondria and chloroplasts. As you know, they have two different energy sources. This is for like the animal part, the mitochondria, and this chloroplast is for the algae and the plants. Unique to mitochondria and chloroplasts is that they divide independent of the cell. They contain circular strands of DNA. Now, where else do we see circular strands of DNA? Correct. We saw circular strands of DNA in the prokaryote. So can you kind of remember that whole thing where we learned about where eukaryotes came from? It was this larger prokaryote that ate up some or engulfed some of the smaller prokaryotes, and those cells became very specialized. Now we know them as organelles, and that's what we're talking about here. Well, here and here and here when we talk about organelles. Well, it's unique to mitochondria and chloroplasts that they divide independent of the cell. They contain circular strands of DNA, and they have a prokaryotic size 70S ribosome. That's 70S ribosome. So that will be pretty important to know. So this supports that whole endosymbiosis theory, doesn't it, of their evolutionary origins, showing that they did indeed come from some type of prokaryotic cell. Let's see what else. So let's go and talk about the chloroplasts resemble mitochondria. Oh, okay. Chloroplasts resemble mitochondria, but they're larger and they contain special pigments and are much more varied in shape when you see a chloroplast. Endosymbiosis theory, I'm going to ask about this. That's why I keep mentioning that, is that chloroplasts were once free living organisms, just like the mitochondria was thought to be, and became part of the modern cell. Evidence is that it has its own DNA, its own ribosomes, and makes proteins, and it can replicate, just like the mitochondria in animal cells, right? Let's go on to thylakoids. Thylakoids are the smooth outer membrane, completely covers an inner membrane, folded into small disc-like sacs that are stacked upon one another into grana. And these structures carry the green pigment known as chlorophyll and other additional pigments. So that's going to be important to know about this inner membrane, the thylakoids, and this grana. Okay? Let's look over here at ribosomes. Ribosomes are distributed in two ways. Now, we've talked about ribosomes quite a bit. Ribosomes are important because some of them make proteins. Now, in our case, in the eukaryotic case, ribosomes are distributed in two ways. Some are scattered freely in the cytoplasm and the cytoskeleton. Others are intimately associated with the rough endoplasmic reticulum. So the rough ER, or the RER, the rough ER, is involved in, you remember, protein synthesis. Good. The S, or the smooth ER, and we know it's smooth because it reminds us of fats or lipids, is involved in making lipids, and that's the non-protein molecules. So make sure you know the differences between smooth ER and rough ER, okay? Let's see, what does my note say? So eukaryotic ribosomes, except in the mitochondria, is larger. So remember, in the mitochondria, it is 70S, just like it is in a prokaryote. And that supports the whole endosymbiosis theory. But in the other parts of the cell, the ribosomes are 80S. And that shows that's a combination of 60S and 40S subunits. These are not additions, because you know that would add up to 100S, but that's not the way it is. These are subunits, so they're not actual weights. So remember, 80S for eukaryotic ribosomes and 707S for prokaryotic ribosomes. You must remember that. And we talked about that difference before on how antibiotics can attack the 70S ribosome, kill that bacteria, but not harm us because we have this 80S ribosome. And it won't harm the eukaryotic ribosome because it's already within a cell being protected in the eukaryotic system, in the eukaryotic system. Let's look at cytoskeleton. Cytoskeletons form a framework to support cells that lack cell walls. That would be animals, correct? This framework, because animals do not have cell walls, so you need a cytoskeleton in your cytoplasm. This framework has several functions, that is, anchoring organelles, providing support and permitting the shape changes and movement in some cells. Two of the main types of cytoskeletal elements are microfilaments and microtubules. Now, microfilaments show the movement of cytoplasm, and some others are active in amoeboid motion that produces extensions, or we call those pseudopods, where they push out and move like an amoeba, right? But anyway, inside of the cytoplasm, you know, those are microfilaments. Microtubules, we saw an example of a cross-section of a microtubule when we cut the cilia. Remember that 9+2 arrangement? Well, microtubules maintain shape of eukaryotic cells that lack cell walls, which that would be animals. They don't have cell walls. They play an essential role in mitosis. If you remember when things pull apart during like in anaphase and telophase, they pull apart. Those are microtubules that you show when you pull things apart when it's in mitosis. They attach to the chromosomes and separate them into daughter cells. They're also responsible for the movement in cilia and flagella. Remember that the cilia had that 9+2 arrangement. That would be an example of microtubules. So, two types of cytoskeleton: microfilaments help with the amoeboid kind of movement in the cytoplasm, and microtubules help with actual movement with the cilia and flagella. So this is an example of cytoskeleton. Okay, so we're back with, I'm going to continue on with the kingdom of fungi, or Eumycota. So if anytime you see mycoses or anything like that, you'll know that that's talking about fungus. So you have macroscopic fungus. Macroscopic means you can see it just with the naked eye. You can just look at it. So you can see mushrooms, something called puffballs, and gill fungi. Mushrooms and puffballs are considered multicellular, so they're complex. They have more cells involved with them. Microscopic fungi will be things like molds and yeast, the little intricate parts of them. So I want you to know that fungi is the umbrella definition or umbrella that has under it molds and yeast. We already talked about chitin is a polysaccharide in the fungal cell walls. Please remember that. Sterols, ergosterols, and in their cell membranes. We have two morphological types. We have hyphae and yeast. Hyphae are the long thread-like cells that make up the bodies of a filamentous fungi or mold, and then you can have that as septate hyphae versus non-septate, meaning, I think they'll look, if you can see this, if you have like cross walls like that, that would be a septate. So you have sections where there are walls, this, or you can have it just plain like this, and that's non-septate, meaning there are no walls. Each one of these will have their own nuclei, and the cytoplasm will not, it can only stay in this compartment, these compartments. Here the cytoplasm can go all around or throughout the septate. So septate has walls, non-septate does not have walls, just remember that. Let's see, so the yeast are so yeast are distinguished by its round to oval shape and by its mode of asexual reproduction. It grows with swellings on its surface buds, which become separate and separate the cells. All fungus are heterotrophic, meaning that they acquire nutrients from a wide variety of organic sources. We're going to talk about that in a minute. Most are saprophotes. I should write that. Saprophotes. Most are saprophobes, and they obtain nutrients from the remnants of dead plants and animals in soil or aquatic habitats. Now, fungi have enzymes for digesting an incredible array of substances. Like we said, they are heterotrophic, meaning they can get nutrients from a wide variety of organic sources. And since they have these enzymes that can digest an incredible array of substances, they can include feathers, hair, cellulose, petroleum products, I should say, wood, and even rubber. Just about every naturally occurring organic material can be attacked by some type of fungus. So fungus is very much easy to find a way to get the nutrients that it needs to survive. Fungus here, I said fungus can be mold or yeast. It's just going to depend on the temperature. At a cooler temperature, 25 degrees centigrade, or Celsius, the fungus is a mold. At 37 degrees Celsius, the fungus will be known as a yeast, right? What was the other word? Oh, pseudohyphae. This is when buds remain attached and they form a chain of yeast cells. So here is hyphae, and then they can look like that, and it'll bud off and bud. That's pseudohyphae, okay? They don't really kind of separate. They just stay kind of budding off of one another. You know, a whole chain of them, whole chain of yeast cells budding off. Let's see what else am I going to say about this one. I want you to remember that fungal spores are used to identify fungus. I think we've said something about when you did your lab write-up, how were fungus identified, and now they're going into this whole DNA thing to identify fungus. But really, the old school way is that fungal spores are used to identify fungus, okay? Sporangiospores are spores produced within a sac. Zygospores, ascospores, and basidiospores. These are all sexual spores. Chitin, remember chitin is in the cell wall of fungus, remember that? Fungal infections are called mycoses, so fungal infections will be called mycoses. Filamentous fungi are known as molds. And remember, some fungus do not have hyphae at all. Some fungus do not have hyphae at all. So I'm just trying to see, did we cover everything for the fungi so we can move on to our last category in the eukaryotic section, known as protista. And I think we did cover everything that I needed to. So let's go to the next kingdom. We move on from this kingdom of fungi, or Eumycota, to now the kingdom of protista. And a protist is a eukaryote that is not a fungus or animal or plant, okay? And this would leave us to algae and protozoans. That is what the protists are. So let's look at algae. Algae are eukaryotic protists that usually are unicellular or colonial, meaning they could be unicellular or they could be in a pack or group, that photosynthesize with chlorophyll A. They lack vascular systems for transport, and they have simple reproductive structures. So algae is pretty simple in that sense. Algae do have chloroplasts, but protozoans and fungi do not. So algae does have chloroplasts. Remember, it looks kind of greenish, so you'll remember that algae is kind of greenish sometimes. Anyway, it has color, and where protozoans and fungi usually are kind of bland, not green usually. Protozoans are unicellular eukaryotic protists that lack tissues and share similarities in cell structure, nutrition, life cycles, and biochemistry. They are microorganisms, and most are motile. And you've seen a protozoan. I think you drew one, that paramecium, correct? And of course they're motile because what did the paramecium have all around it? They had cilia, correct? Trophocytes is the motile feeding stage of a protozoan. Cell walls are not found on typical cells of protozoans, okay? I think, is that it for the, yeah. So here's some miscellaneous things I just wanted to use to close out chapter 5. Let's just look at some things we have here. The first one is endospores, which endospores we talked about a lot. So this is just kind of going over some things. Some things are going to be mixed with chapter 4, because I'm going to kind of just mix chapter 4 and chapter 5 right now, so we could just go over some words and some items that I want you to be familiar with before we have chapter 4, chapter 5 exam. So this is miscellaneous. Endospores, as you know, will go along with protozoans, so it has nothing to do with eukaryotes, but like I said, I'm going to mix them. So endospores, you'll remember the two bacteria that cause endospores would be Bacillus and Clostridium, so make sure you remember that. These are metabolically inactive and resistant to heat, chemicals, and destruction. Resistant to destruction by radiation. Interesting. It's very difficult to kill an endospore. These are living structures. They have calcium diplochonic acid in their cell walls. I might ask that question. Calcium diplochonic acid in their cell walls. For protection of genetic material during harsh conditions is what a bacteria like Bacillus or Clostridium will turn into to protect itself during harsh conditions, right? All right, we talked about thylakoids in chapter, that was, endospores will be in chapter 4. Thylakoids will be in this chapter, chapter 5. This is grana, is a stack of thylakoids. Thylakoids okay, so just remember, grana is a stack of thylakoids. So I'm going to leave it at that. Histones are proteins associated with DNA in the nucleus, right? Histones are proteins associated with DNA in the nucleus. I'm mentioning malaria because it is caused by Plasmodium, and I think you will, that was asked in one of the lab exercises. So when we talk about malaria, which still kills thousands of individuals, hundreds of thousands annually, especially in areas that have a lot of mosquitoes, like in the Mediterranean and in the coast of Africa and off the coast of Africa. So malaria is caused by the genus Plasmodium. Endotoxins. Endotoxins are found in some specific bacterial cell lipids. So if we're talking about bacteria or endotoxins, you'll know that is in chapter 4. That's found in some specific bacterial cell wall lipids and in gram-negative bacteria. Endotoxins cause shock, stimulate fever, and it's involved in typhoid fever and some meningitis cases. That's just a recap, and I know we went over that in chapter 4 already because I drew that chart and I remember that. Helminths, here we're talking about this, that will be in chapter 5 because it's a little parasite. It's a worm actually. It can that parasitic worm. So So just remember that, just remember that's a parasitic worm. A lot of people get those if they're in rice paddies or something, they don't have on shoes, or you walk around barefoot a lot in some of the tropics. The worm can come up through the sole of your foot and wiggle in and stay in your, especially your lymph system. What are plasmids? Plasmids are a circular, round, genetic material. This is seen in the bacteria as well. And plasmids are cool because they can actually change themselves and make the bacteria more resistant to, say, antibiotics, or more resistant to being killed. So plasmids are really interesting. But we use those, the circular DNA, and oftentimes we could cut something out of it and then replace it with something else to make the bacteria get this, and it makes it less resistant. And it can, we can use the antibiotic to now kill it. So we can take pieces out of it and do some modern genetic engineering with this piece of DNA, this plasmid. I'm gonna just draw one more thing. It's gonna be a chart, but I want you to know that this chart should have gone with the chapter 4 with the bacteria, but I put it on a different page in my notes, so I missed it. But it's all chapter 4 and chapter 5 related. But I just wanted you to keep chapter 4 as prokaryotes and chapter 5 as eukaryotes. But since I didn't turn to this page, I missed it. And I just want you to have it in your notes so that you will understand how bacteria grow. So this is going to be a bacterial growth curve that I'm going to draw. And then that will conclude chapter 4 and chapter 5, because I think we would have gone over everything. Okay, so I'm going to draw that. All right, this is something that needs to go in chapter 4 because it's talking about bacteria, but it was in the wrong place in my notes. So let me just go over this and just know that after this will conclude the chapters, the chapter 4 and 5. I'll just combine them since we did 4, now we're finishing up 5, but this part needs to go in chapter 4. So this is the bacterial growth curve. I just wanted to share this with you so you understand something about bacteria. Here, always know your axes, your X and Y axes. So here's the X, which will be time, and the Y axis is the log number of cells there. So we're going to look at the log, in other words, the exponential number of cells over a period of time. So here's a lag phase going all the way up to a stationary phase, coming on down to, let me just start over. Here's a lag phase going up to a log phase, bottoming out or evening out at a stationary phase, and then curve coming all the way down to what we call the death phase. So we'll see as 4 phases, right? Let's go over each one. The lag phase here, this is a period of no growth, really, when the organism is adapting to a new environmental metabolic activity. And this is really without any cell division. So no cell division is going to equal no growth, right? Let's look at the log phase along here. This is exponential. So this describes the steady state of growth, typically at the organism's fastest rate, right here. So there's a lot of cell division occurring along this curve here. And you can see the number over time is going up and up and up. For the log number of cells would be increasing. Here's the stationary phase. You see it kind of leveled off. Nutrients are beginning to become depleted. Nutrient depletion. Growth has slowed. Spore formation in some bacteria. Spores will start to form here because conditions are considered harsh. And remember, the 2 spore formers are Bacillus and Clostridium, correct. So at this point, there's turned into endospores or spores, and they're like, oh, the environment is harsh, I better take some precaution and protect myself. So there's no growth really at this stationary phase. And then coming on down here, shroom, this death phase, this is prolonged nutrient depletion, a buildup of waste products, and of course that leads to the bacterial cell's death. Now, where along on this curve, lag phase, log phase, stationary phase, or death phase, where on this curve would you think antibiotics will be most important or most effective? Where would you think? Okay, so I hear that a lot of you have different ideas. Some say lag phase, some say log phase, some are saying in the stationary phase. Not many of you said the death phase. That's good because they're already dying. But this is where antibiotics will be most effective, will be right here where a rapid growth will be in the log phase. Antibiotics are more effective if the organism is growing. So right here. And over time, this is what you must understand about an antibiotic. When you are prescribed an antibiotic for 7 to 10 days, you should not stop taking the antibiotic just because you start to feel better. Why? Because the antibiotic could need to be the full 7. It has to be the full 7 to 10 day period because it takes that long to actually get a log number of the cells dead. So if you take it for 1, 2, 3 days, here it is here, you haven't really killed them. So the 4th day, maybe, the 5th day, maybe, the 6th day for sure, 7th day, yes. But it's just going to depend on which bacteria that is and which antibiotic you've gotten. Some will kill it by 3 days, some 5 days, some 7, some 10. The point is that you need to complete the full prescription of antibiotics in order to fully eliminate and kill the bacteria. I just wanted to say that because I wanted to share that because I wanted you to see a typical bacterial growth curve, and I wanted you to understand the importance of completing the prescription of antibiotics. So that will conclude chapters 4 and 5. The exam will be on chapters 4 and 5, comparing prokaryotes to eukaryotes, and I hope you enjoy watching it. We'll go into specific bacteria next time I see you. We will be talking about specific bacteria, gram-positive versus gram-negative.